Hi guys, I hope this works. I had a question about running the two softwares for determining membrane protein functionality. I've never used this screen capture before, so I'm going to try to go quick. It only allows 15 minutes recording time. So first thing you want to do is have the bioinformatics guide up, and then you want to go ahead and search for membrane proteins, and that top link that comes up right in this area is going to take you to the page that you want. And I've already clicked that. I'll click it again just so you can see. It opens up with this page right here, and it's pretty much got everything you need. The first thing you see toward the bottom of the screen right now is the link that'll take you to the page, and if I can scroll down, you'll see the link to the second program right here as well. I've already got tabs opened up with those, and I've already run the analysis. I'll go back and walk you through. Let me slide over just a little bit. This is the TMHMM result. And I went ahead and ran gene product 3 for Painter Boy, since they're the ones that asked this question. What this output shows you once you get the output is, for this particular protein, there's no transmembrane domain. The transmembrane domains will be highlighted in red, and you can see that down here. I'm assuming it's capturing my cursor arrow. If you have a transmembrane domain, you'll see it highlighted that way. This one shows that it's all a globular protein. So again, this is the result for the TMHMM results for gene product 3 and Painter Boy. And then I did the same analysis for the SOSUI analysis. And what it tells you is right here in the center of the screen, this amino acid is a soluble, or amino acid sequence is a soluble protein. That again says it's going to be soluble. It's going to be in the cytoplasm of the cell and not embedded in the membrane. So for that one, you would not label it as a membrane-bound protein. I'm going to go back. I've got two monitors working. I wasn't sure I could do this, but I'm going to go back and I'm going to step into gene product 4 now, and I'm going to do the same analysis. So I'm just going to grab the amino acid sequence for this protein from Painter Boy gene product 4. I'm going to go here. I'm just going to click backward. I didn't show you that, but you'll see it on the next step. And if I can get back up to this monitor. Come on, come with me. And I went back to this tab. This is the amino acid sequence for the previous analysis, so I'm going to delete that, and then I'm going to paste in the next one, and I'm just going to hit submit on that. And then while that's running, I'm going to go over here to the other analysis. I'm going to delete out the sequence here, and this one's actually gene product 4, so I'm going to change the name there just so I don't get confused. It allows me to. The other one doesn't allow me to sign a name. I'm going to click here. Again, it says that one's a soluble protein, so I'm assuming this one's going to say the same type of thing. Beautiful. I'm going to go back down to my DNA master. I want to find one that is a membrane protein. This one maybe has some possibility. It's a much bigger protein. So I'm going to go backward, and you can see how quick this analysis is. I'm going to delete that amino acid sequence, paste in the amino acid sequence for, in this case, gene product 5 from Painter Boy. And I'm going to go back in here and just step backward with the back arrow, delete that sequence. Again, paste in. I'm not going to, well, I'll do it just so you know you want to keep track. And so this is gene product 5. I'm going to run it there. That one says it's a soluble protein. This one says it's a soluble protein. I really want to find one that's a membrane protein. I'm going to go in DNA master. Let me drag you down there since this is going pretty fast, but you can see how quickly it goes. This is as large a screen as I can get. I'm going to go feature, and I'm going to go description, and I'm going to go down here to find a protein that actually looks like it's probably going to be a membrane protein. This based on the description that you all have given. Okay, here's the gene that's labeled lysine A. That really should be a membrane protein because it's supposed to punch holes in the membrane. So I'm going to go ahead and grab that and in my clipboard and go back up here and get back into this page now. And I'm going to step backward. I'm going to get rid of all of that. Just delete it out. I'm going to paste this in. I'm going to submit it. And I'm going to go ahead and do the same thing on this one. Step backward, get rid of, and I didn't notice what the gene product number was for this particular protein, but I wanted to find one that would work for you. I don't believe that result. Let me go see. Holland was the next one down, I think. And let me see what's going on here. I may not send you this if it doesn't work right, but we'll find out. whole, and it should really have a transmembrane domain. So if I'm wondering if they actually labeled their lysine A correctly. Painter Boy team, you might want to go look at that. Here's the whole, and it should definitely have a transmembrane domain. There we go. And let me run this one as well. And you know, lysine A may not actually be embedded in the membrane, but look at that. So you get the output here. This one has 2 transmembrane domain regions, and so even if this was the only test that said it was a positive membrane protein, we would write membrane protein as the function. That's the SOSUI. And now if we go back to the TMHMM, it's showing this one transmembrane domain region, and we've got another one that's right here that isn't actually identified well, but they're showing you it's because this one doesn't meet the threshold. So this one's saying there's only one. You can tell it by this bar. The SOSUI says that there's 2 distinct, a primary and a secondary. I suspect it's calling this region the secondary right here, but it doesn't meet the threshold for TMHMM. So if we got this result in TMHMM and didn't get a positive in the SOSUI, we would have to not call it a membrane protein. But this one's indicating there's 1 helix domain, and you can actually see that here, and they tell you which parts inside and outside right in this region as well. And so for this one, we definitely are going to call it a membrane protein. The SOUSI already meets the criteria of 2 transmembrane domains, and the TMHMM tells us that it believes there's at least 1 transmembrane domain. Remember, the criteria are either 2 transmembrane domains identified by one program and none in the other, or at least 1 in each of the 2 programs. If any program, either of those 2, identifies 2 transmembrane domains, we call it a membrane protein. If both just identify 1, we should also call it a transmembrane protein. I hope this helps. Let me know if there's other questions. Thank you.